welcome to our podcast series on synthetic biology.
We're the City of London iGEM team of 2024, and our goal here is to release weekly podcasts you can listen to anywhere at any time on various biological topics.
Today we're talking about prokaryotic cells. So without further ado, let's dive right in.
So, last episode, we talked a bit about prokaryotic cells when mentioning the differences between them and eukaryotic cells, but a little recap is always a good thing. As we said last episode, eukaryotic cells are all cells that aren't bacteria or archaea, and we'll talk about archaea a bit more later. Human cells, plant cells, and protoctista are all eukaryotic cells.
Prokaryotic cells, meanwhile, are bacteria and archaea. Prokaryotes tend to be single-celled, eukaryotes tend to be multicellular.
There are many differences between prokaryotic cells and eukaryotic cells.
So what's the main one?
Well, bacteria have no nuclei.
Do you guys remember what a nucleus is?
Go check out our previous episode for our breakdown of all the organelles within eukaryotic cells if you forgot.
Other differences between those two cell types are that there are no membrane-bound organelles within bacteria. But, what does this mean? Basically, membrane-bound organelles are organelles such as mitochondria, the nucleus, the ER, and Golgi, which we all talked about in episode 1. You might be wondering, why is this? Well, that all comes down to endosymbiosis theory.
What is the endosymbiosis theory?
The theory refers to when one single-celled prokaryotic cell essentially engulfed another to become a more complex organism. And this happened in the origin of life. And this organism that the bacteria engulfed would have had an interesting function, such as energy production or photosynthesis. And since this function helped the bacteria, they had a symbiotic relationship and evolved together, giving rise to the eukaryotic cells that we know and love. So, it makes sense that prokaryotic cells have no membrane-bound organelles such as mitochondria, because these were once the freestanding bacteria that other bacteria engulfed, but obviously with prokaryotic cells that did not happen.
Taking a step back from this, we have been mentioning archaea a couple of times, and this is a group of never before heard of in this podcast organisms called archaea. And initially, everyone thought that archaea were the same thing as bacteria, but actually they're not.
So what is the difference between the two?
The distinction is made through our evolutionary understanding of how living things came to be. Scientists now believe that life originated from archaea and not bacteria.
Talk about all the different types of organelles we find in prokaryotic cells, starting with circular DNA. And it's as simple as it sounds. It's literally DNA in a circle without any of the histones that we mentioned in the previous episode when referring to eukaryotic cells. So this means that there's no condensing of the chromosomes because there's no chromosomes and there is no mitosis.
So wait, what's a plasmid then?
Plasmids are actually also small cycles of DNA, but they are slightly different from circular DNA in the way that the synthetic biologist uses them. Because plasmids are separate from the circular DNA that we talked about, they can be isolated very easily, and it makes it very easy to genetically engineer bacteria. Moreover, plasmids are quite useful because they can travel in and out of the cell, and a great example of this is antibiotic resistance within a culture of bacteria. So that sounds quite weird. Let's break it down. Basically, if you have a culture of bacteria and one of the bacteria gains antibiotic resistance, it can then transfer this plasmid, which has the antibiotic resistance, to other bacteria through horizontal gene transfer. So we can see how useful it is that one bacteria can transfer its plasmid from one to another. And obviously this is something that eukaryotic cells do not possess.
So interesting and quite terrifying too, especially in the context of hospitals. I was thinking MRSA, Do you say MRSA or do you say MRSA?
I say MRSA. Someone converted me. I used to say MRSA, but I got converted.
MRSA sounds better, doesn't it? So the next organelle we're going to talk about are food granules, and they are, again, self-explanatory. They're little vesicles filled with lipids, except they're slightly different because the actual vesicle is made out of glycogen. So if you didn't know, glycogen is a polysaccharide. A polysaccharide is a polymer or a molecule that is made up of repeated subunits, and this polymer is made of repeated carbohydrates. This little bag made of glucose holds these lipids, which are any fats and oils that can be broken down for energy.
That's so interesting. Then we move on to the mesosome. So The mesosome is now thought to be, because of the way scientists prepare bacterial slides, and is now thought to not be a structural part of bacteria serving a function. But, we might think that it's quite similar to the cristae that mitochondria have, like the membranes that fold in and out of mitochondria. So you might think they are quite similar, but I think it's been recently proved that it was actually just because of the preparation of the slide. So,
That's so interesting. We haven't yet talked about the cell wall, though. This is one of my favourite parts of bacteria because it has a cool name. It's made of what's called peptidoglycan. That's such a fun name. So what is peptidoglycan? If we break down that word, we have peptid, which kind of resembles the word peptide. And if you're familiar with any sort of GCSE biology, you know that peptides are describing a series of amino acids. But not only is it made of peptides, we also have polysaccharides, which I mentioned earlier.
Okay, right. So moving on to yet another part of prokaryotic cells, some of them also have a thing called a flagella, which is basically a tail. And the bacteria uses it to kind of move around, to rotate, almost to swim. And I think that is very nice.
One really common example of this is with sperm, because the way sperm swims up the vagina to get to near the fallopian tubes where the egg is through its tail, which, people don't often connect the fact that that's the flagella, but, cool little fact. Then we have the pili, which is often also mistaken for a tail-like structure of the bacteria, but it's actually not. The pili basically allows the bacteria to stick to surfaces. Maybe this has a correlation with infection rate, because bacteria have more pili then they're better at sticking to surfaces, so they are at a higher chance of staying and infecting people or things that touch the surface. But this is speculative. I'm not too sure on that.
Interesting. I've read that the surfaces in which the bacteria use the pili to stick to are a very good predictor of whether or not that culture will form a biofilm. Do you guys know what I'm talking about? So biofilms are a really, really tricky aspect of healthcare management and in infectious disease control. This is because they form these little bubbles in which they can protect themselves from things like antibiotics. So we earlier talked about plasmids and how horizontal gene transfer can help bacteria become resistant to antibiotics, and forming biofilms only helps aggravate the problem.
I see. That's so interesting. I never would have made the connection, but it makes sense.
Yeah, it's incredible. Also, the bacteria have a capsule, which is a slimy layer that covers the bacteria. It helps the bacteria survive in really tough conditions. So the slime gives the bacteria an added layer of protection, but also allows for the bacteria to be hydrated. And I'm not sure what you guys think about this, but do you think at all that it helps with the movement of molecules in and out of the cell? Do you think it helps with the dissolving of gases, similar to the way surfactant in our lungs helps dissolve oxygen into the bloodstream? What do you think of that?
I would think that's really, I would agree with that, because, as you've said, the alveoli also have that sort of thing going on, and we've seen that in humans it does increase the efficiency of gas exchange. So I don't see why it wouldn't be the case in bacteria.
Yeah. But before you believe us, go check it out for yourselves on PubMed. Not on PubMed, just.
I mean, PubMed's good. PubMed's good.
Yes, PubMed's good.
The final thing to say is that it's worth noting that none of the structures that we'd mentioned are membrane bound. So, for example, the food granules that Irene was talking about are made of glycogen, not lipids.
So that's all that we have today, and feel free to leave any questions in the comments and tell us what you found interesting.
And of course, don't forget to check out our Instagram at iGemCol and TikTok, where we share updates on the latest synthetic biology news and all of our adventures.
Thank you for listening, and be sure to tune in next week for our third episode on cell transport.
Bye for now.